Hi, welcome to the session on neurodevelopmental insights from fetal and infant imaging. I'm your chair, Vani Pariyadath, and the session is also being chaired by Dr. Michelle Freund. We're both from the National Institute on Drug Abuse. So in recent years, we've really come to appreciate the extent to which early life experiences impact brain development. And we've come to understand that many of these effects persist well into adulthood in terms of their effect on the brain, behavior, and most importantly, on the risk for multiple neuropsychiatric disorders, including substance use disorder. As you can imagine, there are numerous challenges to studying neurodevelopment. For example, the presence of motion in infants and fetuses during imaging. In this session, we're going to highlight some of the recent advancements, some of the technological advancements we've made in dealing with some of these challenges. And we're also going to discuss some of the highlights. We're going to discuss some of the recent advancements in our understanding of neurodevelopment afforded by applying this technology. And many of these technologies are being implemented in the ongoing HBCD study. And you're going to hear more about that from Michelle.
Thanks, Vani. And if you can control the slides, great. So I am Michelle Freund from NIDA, as Vani mentioned. And the Healthy Brain and Child Development Study, or HBCD, is one of the many initiatives that have been developed as part of the NIH response to the opioid epidemic. In 2018, the NIH received a congressional appropriation of $500 million per year to address this crisis. The Helping to End Addiction Long-Term, or HEAL initiative, was launched with a number of programs developed to tackle various aspects of the crisis. Next slide. The HEAL initiative is roughly divided into two large research areas: enhancing pain management through studies that seek to develop novel, non-addictive pain medications, and clinical research in pain management. The second area is focused on improving treatments for misuse and addiction, including improved prevention strategies and enhancing outcomes for infants and children exposed to opioids. The next slide. Over the past 15 years, the number of infants born with neonatal opioid withdrawal syndrome increased over 5-fold. Early exposure to substances, including pre or perinatally, is linked to greater risk for developing substance use disorders later in life. Prenatal exposure is also associated with other behavioral problems, including ADHD, conduct disorder, anxiety, and other mental health disorders. However, a causal link is difficult to establish due to confounding factors such as socioeconomic, environmental, and genetic influences. Results from the National Survey on Drug Use and Health show some promising news related to substance use among pregnant women. A decline in illicit drugs, tobacco, and alcohol was noted between 2017 and 2018. Nonetheless, the long-term impact of substance exposure in utero remains largely unknown. Next slide. The HBCD study is expected to be a longitudinal observational study that will characterize normative neurodevelopment from birth to 10 years, with an emphasis on providing information on the impact of in utero exposures to potentially harmful substances and the environment. The key research objectives for the HBCD study are to characterize developmental trajectories, including structural and functional development of the brain, revealed through repeated neuroimaging. You can advance through the slide. The study will also establish cognitive, behavioral, social, emotional, and academic trajectories measured with a multitude of assessments over time. These measures will begin shortly after birth and continue for 10 years. Using a multitude of assessment technologies, we hope to delineate the roles of sex, genetic, epigenetic, social, and other environmental factors on both risk and resilience related to brain development, social and behavioral maturation, substance use, and mental disorders later in life. By virtue of its long duration, the HBCD study aspires to assess the impacts of these exposures and the factors that may mitigate these effects as children age, begin school, and establish lifelong trajectories. Only the results of such a long-term study will be able to inform the development of interventions to restore healthy trajectories and interfere with continuing cycles of substance use and mental illness. It is our great fortune to have the set of highly respected speakers in this symposium that will provide their perspective on the importance of using neuroimaging as a tool to illuminate early neurodevelopment and to address some of the challenges and limitations of this approach in this population. Thank you for your attention.
Thank you to Vani for organizing this group, and also to my co-presenters and for the opportunity to contribute to this discussion about advances in fetal and neonatal imaging. Our work is focused on early human large-scale network development and developmental disease origins beginning in the womb. Many years ago, I joined the perinatology research branch of the intramural program of NICHD, and at that time initiated a program of research that was focused on understanding brain development before birth, using techniques such as fetal resting state fMRI, which is predominantly what I will focus on in my talk today. I will describe developmental properties that are normative of fetal brain development, and then I will also share some recent work that we've done examining the effects of maternal stress exposure on fetal neural network development. Now, I think one of the reasons that this field has developed more slowly than other areas of pediatric imaging has to do with the many challenges inherent. But one thing that we've learned over time is that the fetus does alternate between periods of movement and quiescence, and that what we are able to do is utilize those data that were taken during those periods of quiescence and concatenate them together. So yes, the fetus is a moving target, and despite our best attempts, we cannot ask the fetus to please remain still. However, by scrubbing the data and doing rigorous quality checks, we are able to use low motion data. But that's not the only problem. There's no single orientation for image analysis. The fetus comes in any direction. We lack automated atlases or ability to segment these data from the surrounding maternal tissue. We lack registration processes, and even with atlases that will in the future and are now being developed, we need atlases for every age of fetal brain maturation. The fetal brain is also small, so the effective resolution of our voxels is reduced by the nature of imaging a smaller object. And if that was not enough, the fetus is encased within an independently moving maternal compartment, which contributes its own slew of physiological and noise to the images. And then finally, I think one of the most important and least understood of these aspects of the challenges is that we possess limited knowledge of the physiological basis of the bold MRI signal, and that's an important thing for us to tackle in future work. Due to time, I will not spend a lot of time on the analytic processes used for analyzing fetal resting state data, but there are a number of papers that we've published. I'm citing some of them here, and I'm happy to talk offline. Also, my group is very open to data sharing and approaches because we would like to see this field grow. So now to share a little bit about what we've learned about normative brain development, we now have evidence that as a fetus matures, efficiency and network structure increases. Therefore, the time it takes a message to get from one point in the system to another is increased. It requires fewer steps. These local neighborhoods give rise to larger communities across which communication can happen. And this is accompanied by physical outgrowth that we know to happen during this time, where local connections extend to increasingly distant brain regions. We also see that modularity across neural subworks decreases with advancing age. That is, modularity, which is the inverse of independence. So what we're seeing with advancing fetal age is integration of these subnetworks. That initial reaching out and forming connections across the networks, we know that will later be pruned and will become more specialized to the developmental processes that they support. And you're seeing a little bit of this here in this image, where you have in the younger fetuses these very isolated areas that are subnetworks. Later, you see that this green network is extending both from anterior and posterior portions of the brain. So that integration is increasing over fetal age. We've also looked at the hub structure, so the central connectors in the overall topographic architecture of the fetal brain systems, and we see that those hubs do emerge during fetal life, with a lot centered in the cerebellum, which is something that's different than we see later in development and even in the newborn brain. We've also seen that the brain has rich club organization, which means that the highly connected regions of the brain are preferentially more highly connected to other highly connected regions. And so what I'm describing here overall are topographic properties that suggest that there's an existence of a dense and sophisticated interconnected network structure in the fetal brain, even before birth. So this led to a question that we wanted to ask some colleagues of mine from the Netherlands. We were curious if this sophisticated structure exists even before you're born, how much does that network structure look like the network structure we know to be present in, say, children or adults? So we performed an analysis where we looked at the 10, what I'm showing here are the 10% most strong connections across the entire brain, divided into the neighborhoods that they occupy. They're shown here as blue dots. And what you see is sort of a fingerprint of who's most strongly connected to who. Now, if we perform the same analysis in a group of fetuses, so this is a group of adult functional connectivity modules, then how much overlap could we expect to observe? We did not know what the answer would be, but in a paper that just came out at the end of last year, we observed 62% overlap between a group of fetal brains and a group of adult brains looking at overall topographic connectivity. So armed with knowledge that the fetus possesses such an elaborate neural architecture naturally leads to examination of the topic that was raised at the beginning of my talk, which is the effects of prenatal stress exposure. We know that approximately 30% of women have experienced stress or trauma during pregnancy, and that as few as 5% that have psychiatric disorders receive treatment during this time. We also know that there are impacts of stress and anxiety on the child, and this has been seen across brain development, across function of the HPA axis. And in animals, we know that these effects begin even before we're born. So what we wanted to ask was, in a large sample of fetuses, in this case, we were able to utilize data from 118 fetuses. The exclusion criteria for the study included being born before 37 weeks or having a birth weight less than 2,500 grams. Also, the data had to be available for processing at the time that we ran the analysis, because what I haven't described is the very rigorous and manual steps involved in processing these data. So what we were able to use was 118 data sets. I can't remember the actual amount of data that each participant contributed, but I think that the mean was approximately 5 to 6 minutes of resting state data. The analytic strategy we used was to divide the brain using a data-driven strategy, in this case, Cameron Craddock's parcellation, spatial temporal parcellation strategy, to divide the brain into equivalently sized regions that comprised all of the cortical surface and some subcortical regions. We then applied a community detection algorithm to further divide these individual parcels into neighborhoods, or more precisely subnetworks. And this resulted in a total of 16 unique networks in the fetal brain was the best model fit. I'm showing only a subset of those here. We then asked a simple question, which is, does the way in which these networks communicate with one another differ with the stress of a mother? And what we found was stress of a mother was associated with differences in frontal-parietal connectivity, striatal, and also temporal, insular, and motor network connectivity. So here we've confirmed that indeed the stress of a mother is written on the brain of her child even before birth, raising questions as to the meanings of these particular effects. Stress was related to frontal-parietal and striatal connections. These are networks we know that are present in the fetus. We also know that these are networks that are modified in response to trauma. And we know that alterations in frontal-parietal connectivity has been linked to a number of disorders, leading to some suggestion that it may have transdiagnostic implications. Now, as we consider the frontal, motor, and temporal insular systems, what we might think about is that, again, we know that these are areas that are strengthened with advancing fetal maturation. We also can think a little bit about the insula and the special role that it plays in development. We know, for example, that it is a hub from a very early point in human life. And we also know that it has been implicated in generating the signals that may be important for building signals and building neurogenesis within the brain. We also have a project a study that we published a number of years ago showing that this is a region of the brain that shows differences in fetuses that will go on to be born prematurely. So these observations lead to a natural conclusion, which is that it's possible that prenatal variation in these subnetworks connectivity may predispose these children to long-term processing differences across these and other related behavioral domains. So in a way, we've connected the dots. We've shown data that present evidence that alterations of fetal functional connectivity associated with prenatal stress are observable before birth. However, there are some problems, and these problems are difficult to address. The first point I want to raise is that we don't have a strong representation of good versus bad in terms of fetal brain connectivity. This is a tricky thing. When you don't have a lot of context, you don't have a lot of other data, it's difficult to say that because you saw connectivity of a certain kind in the prenatal brain that it had a particular meaning. The second consideration that we have to take into account is that often there are other things that accompany prenatal stress that themselves could have potential impact on how these circuitry develop. So to the first point, we really want to be able to say something about how these stress-related patterns relate to future mental health and cognitive development. We want to know what these fetal connections mean to a child's future self. And for this reason, 9 years ago, we developed a longitudinal cohort. At this point, we have more than 200 children that are between the ages of 5 and 8 that were scanned in the newborn period and in fetal life. And what we will do is we will begin to analyze the relevance of these kinds of differences to future behavioral development. And in fact, we have exciting studies underway right now. To the second challenge, we thought that it was important to look at these factors in this particular data set to try to understand their contributions to the stress brain effects that we observed. And so we specifically chose to focus on 5 different domains of maternal health behaviors, as well as interpersonal health support across 5 domains. And in order to take a stab at reducing the overall number of variables that we looked at, we performed an exploratory factor analysis. And what we observed was that these derived factors fit into 3 factors, some of which seemed most linked to health behaviors, one that was most related to interpersonal support and cohesion, and then finally a measure that crossed both domains. It had aspects that were contributed both from health and both from relationship support. Now what we did is we put these into the model, and what we found was that there was a significant interaction with these particular factors in that stress to brain effect. So I'm just going to break that down for you here. It's actually a little surprising what we found. We found that the stress to brain association was stronger in the mothers that were overall healthier. So this might be a little bit different from intuition that would suggest that the supportive relationship would buffer the effects of stress. Instead, what might be happening is that these behaviors may be loading on these same brain circuits such that those that have overall higher stress have less good overall health, and then there's less opportunity for change within these networks. And if we look at what specific factors played a role, I'm listing them here. And what we also noted was the extent to which those factors contributed varied across what we looked at. So I think what's exciting about these data is that it amplifies that these stress brain associations can be modified behavior. And this is an exciting thing because we want to think about pathways for intervention. And it may be a little bit more challenging to address something like stress, but maybe if we understand that sleep or interpersonal support play a strong role in these associations, those may be places where we can intervene. So in a final question, we asked if the magnitude of the effect of stress on the fetal brain related to prematurity. Did that stress to brain effect predict how early a child was born? And it did. In other words, the stress of the parent raises the likelihood of premature birth in addition to potentially modifying that child's brain. And since we already know that being born early sets a child at a disadvantage, we now know that that baby has 2 challenges. And you will note that the association between the network connectivity and the strength of the association between the age at which a child was born and their connectivity does differ across different brain regions. So we can also begin to dive into those effects specifically. So I'll close here. We're armed with evidence that the fetal brain experiences maternal stress. And as we begin to think about optimizing growth and resilience, the ideas that this project helped me to nourish are that it may be difficult to tackle, as I said, topics as big as stress of the mother, but perhaps there are available potentially modifiable factors in her life where we could push the needle on her health and therefore affect that developing child. Another point I want to raise is that biology could help us in this path. So in this study, we were able to look at the impact, the magnitude of the impact of the stress on the brain, and those connections themselves told us about additional aspects of the mother's life and also the health of her child, or when her child will be born in this case. And so as we continue to follow these children, I think what we need to do is to build up data sets that will longitudinally evaluate human development so that we can, as I pointed out, better contextualize these factors. Because ultimately what we want to know is how, in a world of increasing risks, we can offer children and families the pathway to happiest, healthiest futures. And also noted here is the need for large-scale multi-site studies, because in such an exploratory space as fetal and neonatal imaging, the need for reproducibility is and replication is incredibly high, because we want these to be more than exploratory studies. We want them to be confirmatory studies that help us to take away what it is that we know is contributing to healthy human development. So I'll close by thanking the amazing team in Detroit that has been continuing to collect this data, also critically our funding agencies, and also a thank you to the many families that over the years have shared their children's development and lives with us. And also thank you for your attention.
Thank you very much for inviting us to talk today. I'm here on behalf of my colleagues in the Developing Human Connectome project, Joe Haenel, Steve Smith, and Daniel Ruckert. And we want to tell you about an open science project, which we hope you'll all engage with in the future. So the Connectome project is an attempt to make a comprehensive mapping of the neural connections in the brain. It's a grand challenge for neuroscience. I think we're all aware that it's not a simple concept, but it's a very powerful one. And it's particularly powerful because we can use network mathematics to look at the macro structure, the millimeter-sized structure of the brain. Our aim is to make the first map of connectivity in the fetal and newborn period, in utero and ex utero, to include normal and abnormal children in it. And we want to make it possible for scientists to address some critical questions about how connectivity assembles itself, what genetic environmental influences might be on that connectivity, and whether we can help to try and explain some of the more intractable neurocognitive and neurodevelopmental disorders which the human race is afflicted with. We started off by rethinking how we image babies. We thought we should try and start from the beginning. So we developed a completely new mobilization and imaging system. We had a new tray for the baby to lie on with a new acoustic shield, a 32 receiver coil set up inside the environment. And we developed a new method of aligning the baby in the coils. The new setup had a little helmet, rather like a motorcycle helmet, which the baby fits into. And that helmet fits directly into the coil, giving perfect alignment. So we slide the baby into the helmet and then slide the helmet into the coil. And that gives us much better alignment and proximity to the imaging coils and consequently better imaging. And this arrangement, which looks incredibly simple, has actually improved our signal to noise ratio significantly. And this is the signal to noise ratio from our imaging coil, the neonatal imaging coil, and imaging setup, compared to the normal adult coil, which we used to use. This has been put together with a lot of help from the radiographers in our department, led by Ian Hughes. And we get results which I think are pretty good. So 0.8 millimeter resolution. This is the T2, T1, and MP RAID scans from the program. I hope you'll agree, these are pretty high quality images. Most of our children get images of this quality. We've had one small problem, which is that because of the very tight parameters we put on the system, some of the babies have actually wriggled a little bit, and we've lost a couple of slices in the brain, which is a shame, but actually, in general, the images are of this sort of quality. We've also had to rethink our diffusion imaging. So Donald Tournier and Joe Haenel have put together a lot of work working out how best to do diffusion imaging in our babies. And we've come up with a system which has 300 directions at 4 shells and produces really high quality data in this population. We run a lot of images in our hour and a bit that we do the imaging in. We're taking an image roughly every about 27 images a second. And the success rate's been a lot higher than we've been used to in the past before we began our redesign of the imaging setup. Previously, in term babies who were liable to move a bit, we only had about 50% success rate in getting high quality imaging from them. But now with the new setup, we've got about 91% success rate, which I think anybody who does this kind of work will recognize as being pretty good. So we also want to image the fetus. And of course, the fetus has this one very obvious and particular advantage, disadvantage, which is that you can't stop it moving. The baby will move around inside mum, and you can see the kind of motion we're having to deal with here. And this is profound motion, which is enough to wreck any kind of conventional MR scan. So we had to develop motion correction to deal with this. The way MR works, the in-plane resolution is pretty good because the scans are fairly fast, but it's the alignment of scans to make a 3D volume, which is a particular problem. And you can see here, the in-plane resolution of this particular scan is okay, but as soon as you line them up to try and make the 3D volume, you get very seriously corrupted data. And it's because the baby's moving inside the scanner coordinate reference frame. So if we take a slice like this, it starts being there. Then the baby moves, the reference frame stays the same, but the baby's in a different place. So the lines are all different, and we don't get a nice, neat lineup of scans. However, we can take these scans and we can oversample in a different direction. We can then use slice to volume reconstruction to put these back together again. And this gives us the opportunity to use super resolution, and with outlier rejection and intensity matching, we get some pretty high quality data from this. And this is a result of slice to volume reconstruction of the type used in the Connectome project. So the top three pictures, you see the pictures you saw before, I think. The in-plane resolution is good, but the reconstructed volume is corrupted. The lower three pictures, you can see the reconstruction using slice to volume reconstruction. And we've got really high quality data coming from this. And using this kind of data, we've got 3D information that we can start to use for neuroscience. It's no surprise that actually babies move as well, even when they're not inside their mothers. And this method's actually been very useful for improving the quality of data that's come out of the neonatal scans. And it's one of the reasons why we have a high quality, a high percentage success rate in neonatal scanning. We can use it on fMRI using the same principle of taking slices, which in the scanner are aligned but misaligned relative to the baby. We can realign those slices, get the fMRI slices together, and get high quality data from those slices. And this is a comparison of fetal fMRI scans to neonatal fMRI scans of babies of the same age. The fetal scans are on the left of these panels, and they compare reasonably well to the fMRI we were doing in babies a few years ago, well, a decade ago now, and produce data which we should be able to use. So fMRI can also be motion reconstructed to get data from the fetus. And without laboring the point too much, you can do the same with diffusion. Here is the original diffusion scan on the left, then the corruption caused by the baby's motion. This moves the diffusion tensor, which can then be corrected using the equivalent of slice to volume, and we can produce tractography from the fetus using this method. So with these methods in our work bag, we could go to work collecting data from babies. So we aim to collect compatible neonatal and fetal images from babies between 20 and 44 weeks of gestational age, both normal and abnormal. We wanted to get an enrichment for people who were at high risk of neurocognitive disorders, because we're interested in the origins of neurocognitive disease. And we collected collateral data to go with this to enable the images to be used for different studies. So we collected genetic data, clinical data, social data, and we had 18-month outcome data using the Bailey-3, a series of developmental questionnaires. And we also invested in gaze tracking because we felt this was a useful way to get a lot of information quickly from our relatively large cohort of children who had to be studied quite rapidly when they came back. So recruitment, we have a total of 1,163 participants. Of these, we have 963 who had at least one usable scan. This means the total number of scans for the infants and fetus that we took was over 1,400. Now, you can see that we did a lot of development scanning. So almost 300 of these scans were used for technical development. And that means they can't be used for data later on, of course. But we have 891 neonatal scans, of whom about 150 were preterm babies scanned preterm, and 150 preterm babies scanned at term, and nearly 300 fetal scans. At the moment, the follow-up is paused because of the COVID crisis, but we're running at 80% follow-up. We've followed up nearly 599 infants. They almost all have pretty complete data sets. So we're expecting 80% complete follow-up or thereabouts when we finish the project, when we can start to see them again after the crisis is hopefully over. This is a bar diagram showing you the distribution of gestational age of birth of the babies. You can see a preponderance of babies born at term. This was planned. And this is the age at scan of the babies. The lighter color bars are the fetal scans, the darker color bars are the neonatal scans. And you can see there's an overlap, but of course we don't go down to the very bottom age range in the neonatal scans. And they come from this area around London in the UK. I'm sorry it's so parochial, but you'll be glad to know that the ethnic and social diversity inside this population is very large. So it's not all the same sort of human being. So what do we do with the data? Well, the first thing that we can do is produce some normal data ranges. So using Gaussian random process modeling, we can produce normal ranges for volumes and other variables. This graph shows the normal range from the population for volumes of various different brain regions with deviations from the normative mean in numbers of standard deviations. And you can see that our population naturally fits into it because these are normal term babies from whom we've created the values from. The value of this kind of approach is that we can now place any single individual on these normal variables for all these different different metrics and effectively do personalized medicine or personalized research looking at individuals, and we no longer have to study groups in quite the same way that we used to. This is the normal absolute volume growth of the brain from about 36 to 46 weeks of gestational age. This is a plot showing the comparison of this normative range to two groups of preterm babies, a group of preterm babies from the DHCP itself, but a group of preterm babies from another large study of 500 preterm infants studied in the E-Prime project. You can see they overlap the normal ranges to a large degree in some areas, but there's significant differences in many of these babies in the size of the ventricles, no big surprise, and also in the size of the thalamus, again, no big surprise. The reason I show this data is to demonstrate, I think, that the DHCP data is compatible with other cohorts when used in the right way. And the graph showing extreme negative and positive deviations is merely to show you that most of these preterm babies have normal sized brains, but there are a significant number of them where it's either too big or too small. If you do group analysis, it's difficult to show this, but we can now do individual analysis thanks to the DHCP, looking at individuals and how they compare to normative ranges. And the same is true not for just volumes, but also for diffusion metrics. This is the diffusion normal ranges done in the same way. And you can see the change in FA and MD in the cerebellum, the putamen, and the frontal periventricular white matter in this group. And you can see that they're actually quite different over time. The rate of change over time is quite different, with a rather complex development in the frontal white matter compared to relatively simple changes in the cerebellum. But again, we can plot individuals against these normal ranges now and see how they're developing. This is the kind of diffusion tractography that the DHCP produces. This is constrained spherical deconvolution using Donald Tournier's methods, showing the cerebellum and some of the white matter tracts. We have all the diffusion metrics, of course. This is the DHCP diffusion pipeline put together by Matteo De Bastiani, and we show values for fractional anisotropy and various other diffusion metrics. These are all available and will be downloaded for people to use. Fetal diffusion tractography also works quite nicely. This is work by Sian Wilson, who's about to present it later in the meeting, so I won't steal her thunder. But you can see the diffusion tensors and the reconstructed corticospinal tracts at different ages here. And this has produced some really quite interesting and surprising results, which I think Sian will be able to speak more about. We have cortical diffusion fMRI maps. These are the surface maps for fMRI activation done by Emma Robinson and her colleagues. And we have the cortical T1, T2 maps, which people call myelin maps. That may be slightly exaggerating their specificity, but probably measures of cortical lipid. I find these particularly interesting. I think it's amazing how much change there is between the time we think as the beginning of normal term and post-term period, so 38 to 42 weeks. Massive amount of change in the amount of cortical lipid going on in these babies, and actually slightly more cortical lipid at 45 weeks than in the adults, which speaks, I think, of later neuronal pruning. And this is the parcellation, the resting state network parcellation done by Group RCA. Again, Michael Ayre is going to speak about this later in the meeting, so I won't steal his thunder. But these are very precise maps, and he also has some very interesting data. All these different forms of mapping the brain give different results in different ways at different times. And one of the attractions with having large data sets is the ability to put this together. And I think we've taken a leaf out of the work done by Arvind Rasnahan and Jacob Selwitz and people like that, who've done some really interesting work with multimodal similarity mapping. So I'm going to show you just a few results now from not the individual data sets of the DHCP, but of adding them all together to produce multi-similarity metrics. So we have data on myelin, cortical thickness, diffusion metrics, the NODI metrics, and we can make cortical regions of interest with those and see how they relate together. And you can see some examples of that here with a heat map showing the comparison. This is a parcellation done by Daphne Finchel, who's just got this published in Cerebral Cortex, showing a multimodal cortical parcellation, which actually is quite interestingly precise and has very clear boundaries. These parcellations that she's produced can be looked at in terms of their coordinate growth. And we can see areas of brain which seem to be growing together or growing in a similar way. And of particular interest to me is the relationship here between the cingulate area parcellation and the limbic system, which both seem to be growing together, which one would expect from the biological background to this, and other areas which are similar or dissimilar. So we can learn a lot, I think, about brain growth from this approach. We can also compare it to histology atlases. So this is the comparison between the morphometric similarity clusters that we have from our analysis of the DHCP and the digital von Economo atlas, which is available. And there's actually quite strong overlap between the Economo classes and the morphometric similarity clusters in this analysis. It's surprising, I think, just how much overlap there is, given that the Economo atlas is an adult brain and this is neonatal brain. But nonetheless, it's interesting to see how they compare. Not everything compares precisely, as you can see, but the limbic system, for example, is really quite precise. These parcellations can be used to compare to other things, and one of the attractions that I think of data sets like this in open science projects is the ability to combine them together. We've combined the DHCP data set with the BrainSpan genetic data set, and Gareth Balls led this work. And he took the BrainSpan atlas, he found the parcellations in the post-mortem fetal MRI and registered those to the DHCP and produced some regions in the DHCP atlas which were coincident and contingent with the BrainSpan atlas. He was then able to compare these with the genes that were being expressed in that atlas. And he's got some really interesting data showing gradients of gene expression and particular types of gene classes in different brain regions over time, which have real biological sense to them, and I think will be useful for us in the future. So that's an overview of the DHCP. The essence of it is an open science project. Our measure of success will be gauged by how many people use the data. We would love you to use the data. You can go to the website of developingconnectome.org. You can download the first data release now, and very soon the entire data set will be available. It will take a bit of time before we can get all the data, genetics and the follow-up data out on the web, but we'll do it as quick as we can. And we would love people to use this and feedback to us. We've been very gratified by the kind comments we've had from people so far who've used it, who I think find the data reasonably interesting. And we'd love more people to use it. So please go to the website and download it. And of course, I couldn't do any of this work myself. This is all the list of people who've been involved in the DHCP family, my senior colleagues, Joe Haynall, Steve Smith, Daniel Ruckert, but the very, very skilled and talented students, postdoctoral fellows, scientists, psychologists, and other specialities that we needed to do this project listed here. And I thank them for giving me the opportunity to come here and talk to you about this today. Thank you very much.
So thank you for the opportunity to share our results on the imaging of non-sedated pediatric subjects 0 to 5 years of age. So these are my disclosures. Since the majority of the results that I'll be presenting today were collected through the Baby Connect Home project, I'd like to acknowledge the Baby Connect Home research team. All the names that are highlighted in yellow are the multiple PIs, and the name with the underline, those are the study coordinators who worked tirelessly and working together with the family for the recruitment and imaging studies. I would say without their dedications and effort, it would be extremely difficult for these studies to be successful. Obviously, all the team members has contributed substantially to this study, and this is really a collaborative project between UNC and University of Minnesota. So as I mentioned that this Baby Connect Home project focuses on typically developing children from 0 to 5 years of age. We use an accelerated longitudinal design for a total of 500 children. All subjects underwent MRI imaging, and this is just one example kind of to share with you from a different project. This is a child that were imaged starting from birth and longitudinal to about 5 years of age. You can appreciate how much of the anatomical structure changes and also the size of the brain changes. Also the cortical thickness changes also shown here. These are spatially non-uniform changes with cortical thicknesses, which I will spend a little bit more time on the cortical thickness later. Here shows the anatomical postulations that can be used to characterize in early brain development. So as I mentioned previously, we use an accelerated longitudinal design. Basically, our cohorts were divided into 4 different groups. And depending on when the first visit was, they've been divided into different groups. For example, in the A group, the subject were further divided into 3 subgroups. In the A1 group, the subject came at the first visit at birth. For the A2 group, the first visit was at 1 month old and 2 months old for the A3 group. And all these subjects in the A group, they have a total of 5 visits and 3 months separating 2 visits for a total of 12 months. So with this approach, it allows us to be able to cover the 0 to 5 years of age within a relatively short funding period. We have one more group, which is the cross-sectional group, which is not shown here. So the Baby Connect Home project collects anatomical images, including high-resolution T1 and T2 weight images, resting functional MR, as well as diffusion tensor weight images with multiple p-values. So with multiple p-values, it allows us to be able to quantify and characterizing the microstructural changes of white matters. Together with very comprehensive assessment, including the behavioral assessments as well as parents' report, it allows us to be able to quantify the potential relationship between brain and behaviors. For example, using the anatomical image, we can characterize in different anatomical attributes such as cortical thickness, surface area, for example. With diffusion tensor weight images, we can characterize in the white matter maturation and functional connectivity from functional resting functional MR. So I thought that we wanted to kind of share with you some of the lessons that we have learned in terms of imaging non-sedated children. Here, the plot on the left shows how long does it take for a child to fall asleep. As I've indicated, most of the study was done during the time when the child was asleep. So you can see that, not surprisingly, that for younger children, it doesn't take long for them to fall asleep. But the time increases as child ages. So you can see that about 1 year old, beyond 1 year old, it takes about 100 minutes or more for a child to fall asleep. And so this is important in a way that when you design a study where that you want to schedule enough time for a child to fall asleep. Otherwise, you might not be able to get images. In addition, here on the plot on the right shows the how long a child can tolerate the imaging study with a function of age. It's very apparent. Pretty much all children can tolerate about an hour or so of imaging time across all age. So that means that you can design the imaging protocol roughly for an hour to acquire different imaging sequences. Here shows the success rates. Again, not surprising for children at 0 to 6 months old, since most of them sleep most of the time. So the success rate is extremely high, it's about 85%. But then start to decrease as child become older. Particularly around 3 years to 3.5 years old, the success rate is actually about 52%. So this is important in a way that when you consider sample size calculation, you have to take into consideration of the success rates here at different ages, particularly for longitudinal studies that for older children, the success rate might drop. Some of the reasons for unsuccessful scan, most of the major reason actually is the child never fell asleep, and this is about 42% of our unsuccessful scan. And a lot of time when a child came to the imaging center, because it's a new environment and there are just so many things to attract their attention, and therefore they never wanted to fall asleep. Other reasons including kind of transportation from the prep rooms into the scanner room, or the preparation into the scanner, as well as kind of the first scan, when the first scan starts. And also the other thing that we found out was that for children that used to sleep on their side or on the stomach, it's actually more difficult for them to sleep on the prone position, supine positions. So this is something that needs to be considered as well. With the ability to acquire high quality images, how our team has developed novel image analysis tools to allow us to extract the anatomical attributes for characterizing early brain development for different parameters, for example, cortical thicknesses, surface area, convexity, curvature, sulcal depth, and local gyrification index. In the interest of time, today I will be only focused on the cortical thickness. But I want to share with you in terms of the global trajectories, for example, looking at total brain volume, white matter volume, gray matter volumes, as well as some of the other parameters. It's very apparent most of the parameters showing a very rapid rise during the first 2 years of life, and starts to become a little bit slower after 2 years of life. And this is important because in a way that if one would wanted to understand early brain development, particularly during the first 2 years of life, one would have to sample very densely during this critical time period in order to be able to capture these dynamic changes. More importantly is about cortical thickness measurements. As you can see here, it rises up very rapidly during the first year, but then in transition from rapid development thickening into become thinning process after the first year of life. So again, to capture these dynamic processes, one would have to densely sample during this critical time period in order to be able to characterize in these complex developmental processes. In addition, we also characterize in the gender differences. The blue lines here indicates boys and the red lines here indicate girls. So you can see that there are gender effects for most of the parameters with the exception of the cortical thicknesses. Here shows the temporal developmental patterns of cortical thickness throughout the first 5 years of life. There are several important features here. Just to name few, the cortical thickness at the central sulcus area, for example, precentral and postcentral gyrus, showing a thick cortex together with insular areas as well. But about 3 months, 4 months, you can see that these precentral postcentral gyrus area become thinner, while the insula continues to become to be a thick cortex throughout the first 5 years of life. Also, that there are different patterns in the lateral surfaces versus the medial surfaces. As you can see, the medial surfaces here start when the thick cortex in the superior frontal gyrus, as well as the mid and anterior cingulate cortex tends to be the thick cortex throughout the first 5 years of life. So if we look at the rate of change, it provides even more information for us to learn about the developmental process of the cortical thicknesses. From lateral surfaces, it appears that there are two phases of cortical thickening process. During the birth to about 2 months, you can see the first wave of cortical thickenings and start to slow down. In fact, actually, as I mentioned previously, the precentral and postcentral gyrus area start to thinning actually about 4 months old and continue to about 6 months old. Similarly, in the superior temporal gyrus area, also showing the hint of thinning process. This is where the auditory cortex is. Again, showing very early thinning process. Similarly, in the medial surface, the V1 area, primary visual cortex, also showing the thinning process very early on during the first year of life. And as we know that cortical thinning has been implicated to be reflecting the synaptic pruning, which is synaptic pruning also being known as to be associated with functional spatialization. So it shows that the basic brain functional areas such as premotor, precentral, postcentral gyrus, the auditory cortex and visual area showing actually pruning process much earlier than previously has been reported. Also, it's interesting that in the lateral surface, there's a second wave or second phase of thickening process starting from about 7 months until about 9 months of age. Notice that very different from the first phase, which kind of showing the global increase in cortical thickness. At 7 months to 9 months, the thickening process appears to be more on the frontal lobe and temporal lobes area. It starts from the Wernicke's area and Broca's area and migrate toward anteriorly into the prefrontal cortex as well as into the temporal pulse area. But if we look at the medial surface, it's actually very different compared to the lateral surface. There's no such a distinguished patterns about two phases, but rather about one single phase of cortical thickening process. Particularly, you can see that during about 7 months, 9 months, the prefrontal cortex area, anterior cingulate cortex area all become thicker and migrate superiorly into the inferior superior frontal cortex area. So this is very interesting in a way that different times that cortical thicknesses occur and undergoes a thinning process at different time, suggesting that to better understand the potential implications about cortical thickness in relationship to behavior and cognitive development, one would have to really sample densely to be able to capture this cortical information. From a functional MR point of view, this just shows some preliminary results in terms of from a graph theory point of view. As one would anticipate that small wellness increases during the first 2 years of life, and similarly for global efficiency. And local efficiency increases and a peak around 382 days and start to decrease. This is, has been consistent to the data being reported in the literature. Finally, from the DTI point of view, given the fact that we use the 6 gels to be able to characterize in the microstructures, so we are able to calculate per exon and anisotropy coherence, intracellular volume fraction, extracellular volume fractions, and some of the microstructural information to better characterize in the highly dynamic and complex microstructure development during the first 5 years of life. So finally, just to conclude, why it's a daunting task, imaging non-sedated children during early infancy and during 0 to 6 years of life is feasible. Building a strong relationship with the families is the key to have a high retention rate. As we have shown, children at 12 to 36 months old take a longer time to fall asleep and with a lower success rate as well. And most of the children can tolerate about 1 hour of imaging time. It's important to realize that also dedicated image analysis tools are needed to analyze the captured MR images. And more importantly, multiple imaging sessions during the first 2 years of life will be needed or critical to capture highly dynamic and developmental features. Thank you very much for your attention.
Hello, I'm Cynthia Rogers from the Washington University Neonatal Developmental Research Program, and I'm excited to present our work today focused on early life adversity, neonatal brain connectivity and early childhood psychopathology. I have no relevant disclosures. So as we know, there are many psychosocial risk factors that increase risk for childhood psychopathology. And our lab has been really interested in understanding how these same risk factors impact the neonatal brain and continue to impact brain development over time. And while there are several domains that are involved, we've been particularly interested in poverty and its role in the neonatal brain. So today I'm going to discuss our work with the Wonder Preemie study that I lead with Chris Miser. This study was started in 2007, and we enrolled both preterm and full-term children. And at term equivalent, they were placed in the MRI scanner where we obtained images of the neonatal brain. They came back at ages 2 and 5 for social, emotional, and developmental assessments. And they're currently coming back at ages 9 to 10 for another MRI scan and further assessments of their development and psychopathology. So in addition to those outcomes, we also assessed a whole host of predictors longitudinally. But again, today I'm going to focus mostly on socioeconomic status and particularly poverty. While we are interested in a range of outcomes, we've been particularly interested in psychiatric disorders of childhood that preterm children are at greater risk for, including ADHD, anxiety disorders, and autism spectrum disorders. And along with that, we have been interested in particular brain regions, subcortically the amygdala and the striatum, and then cortical areas including the medial prefrontal cortex and the posterior cingulate cortex, major hubs of the default mode network, and then also interested in the white matter tracts that connect these regions, including the cingulum, the uncinate fasciculus, and the anterior limb of the internal capsule. And our lab particularly uses probabilistic tractography to assess white matter tracts, as well as resting state functional connectivity. So I want to start with just reviewing some of the findings we've had looking at longitudinal associations between brain connectivity and childhood psychopathology, starting with white matter microstructure. We've previously published relationships between the neonatal cingulum using the diffusion measure of fractional anisotropy and early childhood social skills and attention. More recently, Rebecca Brenner, a talented MD PhD candidate in our lab, has extended this work and looked at the neonatal cingulum using another diffusion measure, ND, which is inversely proportional to fractional anisotropy. Again, looking at autism symptoms using the SRS, as well as ADHD symptoms at age 5, and continues to find a relationship between neonatal white matter microstructure of the cingulum and these outcomes. Further, Rachel Lean, a developmental psychologist in the lab, has recently been looking at these outcomes with our 9 to 10-year data and has similarly found relationships between the neonatal cingulum and inattention and hyperactive symptoms, as well as some of our task-based measures, looking at the diffusion measures of the anterior limb of the internal capsule, which is involved in frontostriatal connectivity. And again, has found relationships both at age 5 and age 9 to 10 with these measures. Similarly, with functional connectivity, we've looked at the neonatal amygdala and childhood psychopathology and looked at those associations across time, and previously published that amygdala connectivity with the medial prefrontal cortex was related to age 2 anxiety symptoms, and at age 5 we find a similar relationship. And with amygdala connectivity in the posterior cingulate cortex, we found relationships with attention at age 2 and with ADHD symptoms at age 5. So as I mentioned earlier, we're really interested in understanding if particular risk factors related to poverty are adversely impacting the neonatal brain, and maybe why we're finding these relationships between neonatal connectivity and psychopathology. So we've been really interested in neighborhood poverty specifically and have used the Area Deprivation Index, which is an index that comprises 17 various factors and gives one percentile rank, with a higher rank indicating more neighborhood adversity. This was developed by Amy Kind at Wisconsin. So not surprisingly, we have found relationships between prenatal neighborhood poverty and childhood impairment across a range of ages and domains, including behavioral exuberance, which is an early externalizing symptom, age 5 externalizing symptoms, and also executive function skills at age 5, as well as preliminarily age 9 to 10 internalizing symptoms. So talented future medical student, Bruce Ramphal, who worked in our lab, was interested in understanding how this neighborhood adversity was impacting neonatal functional connectivity. And he did find relationships between striatal connectivity, amygdala connectivity, and medial prefrontal cortex connectivity, and the Area Deprivation Index, as noted here, across various regions. But Bruce extended that work and showed that not only is there a relationship between socioeconomic disadvantage, including the area deprivation index and factors like behavioral exuberance and age too, but that this relationship is mediated through the connectivity measures we found, particularly in the striatum and socioeconomic disadvantage. So this is our first mediation relationship, and we hope to extend this to some of the other connectivity findings. In that vein, Dr. Rachel Lean, who I mentioned, who's a developmental psychologist with a focus in executive function, has found that the left anterior limb of the internal capsule diffusion measures are related to area deprivation. And further, that the same measure of microstructure of the ALIC is related to an executive function impairment. And that the area deprivation index, as it increases, you have worsening executive function. And so taken together, this is very suggestive, again, that this prenatal neighborhood poverty is altering the neonatal brain and leading to increased impairment in executive function. So to extend that, looking at age 9 to 10 childhood executive function, we found a relationship between this executive function score and the default mode network connectivity. This is with a talented student that worked with us. And as we see with executive function increasing, you have increasing DMN connectivity. But with worsening area deprivation score, you have decreasing default mode network connectivity, indicating again that area deprivation index is altering the brain and leading to impairment in childhood executive function. So while the WONDER study has shown us that there's evidence of neonatal connectivity predicting outcome, and that psychosocial risk factors like neonatal area deprivation may be the cause for that alteration in brain connectivity, we, along with several other labs, including Joan Luby, Barb Warner, Dana Barch, are working on a study called the Early Life Adversity and Biological Embedding Study, which is a large study of almost 400 infants, where we're really going to be able to assess psychosocial adversity much more in depth, as well as a whole host of other factors that impact social and emotional development. So just some early findings thus far, we do see differences in several cortical networks, depending on whether the child was exposed to high or low socioeconomic status prenatally, and then also finding similar findings with diffusion measures here with the onset, but also we found some findings with the cingulum as well. But perhaps the most innovative and interesting analysis we've done thus far with this study has been led by our colleague Chad Sylvester, who's been interested in understanding early risk factors for anxiety in childhood, and utilizing a task-based paradigm similar to an auditory oddball in the scanner with the neonates. He found that regional neonatal neural activity to those deviant sounds was more robust in the infants who were exposed to higher levels of maternal anxiety. And interestingly, these are the same regions that adults with anxiety have when they respond to an auditory oddball, and are also the regions that are implicated in the pathophysiology of anxiety disorders. So we think we're going to have some very interesting findings from the ELAB study, and so more to come on that. So I'd like to just acknowledge all of the staff in the Wonder Lab who make these studies possible, as well as our collaborators and the families and children who participate in our studies. Thank you.